Hello friends and my dear students. Myself Komal Walde from RBNB College, Sri Rampur. Today we are discussed about the chapter from 11th standard. The chapter name is Cell Division. You know that, students, what is the cell? It is the basic and structural unit. We can say that it is the structural and functional unit of the life, that is cell. In a life, the importance of cell is very much because the cells means the body is made up from the all different types of cell. Cells combine and form a tissue, tissues combine form an organ, and different organs in the life process or in system, and all systems are collectively present in the living organism. So the cell division, it is the stage or it is the process in cell life, because new cells are not formed. The new cells are formed. These are from the pre-existing cell. In the cell theory or in the cytology, the cells are arises from the pre-existing cell. It states that the new cells are arise from the pre-existing cell, and the multicellular organism is developed from the single cell, and that single cell is the zygote. So the importance of the cell division is very much because all the cells are present in our body. It divide and form new cells by the process of the cell division. So all these cell division means cell division takes place in the sequential events. Those sequential events are in such a manner which is known as cell cycle. All the sequential events are occur in cell cycle, and in this cell cycle the development of the cell as well as the division of cell is carried out. So in the cell cycle there is two phases. One is the interphase and another is the M phase. Interphase is a resting phase of cell in which the development takes place or the cell is active in the interphase for further division. Interphase is the phase between two successive cell division, that is the interphase. It is resting phase of the cell in which only development and growth of the cell is carried out. Hence, now interphase is divided into three subphases. We are moving on our topic. Interphase is divided into three subphases: G1 phase, S phase and the G2 phase. So friends, in a cell cycle, G1 phase, S phase and the G2 phase, it is known as the interphase. It is known as the interphase. Okay, so G1 phase. G1 phase is a first gap phase, or we can say that it is the first growth phase or the gap period of the cell, in which growth takes place, in which the cell's growth takes place. Synthesis of RNA is carried out in the G1 phase. Synthesis of mRNA, tRNA and rRNA is carried out in the G1 phase. It is the first growth phase. Again, in which protein synthesis is also occur, and the duration of the G1 phase is nearly 8 hours. The duration of G1 phase or the growth phase is nearly 8 hours, and it is followed by the second phase, that is the S phase. S phase is known as the synthesis phase. S phase is known as the synthesis phase. The duration of the S phase is 6 to 8 hours. In this phase, the synthesis of genetic material, we can say that the DNA synthesis is carried out in the S phase. The duration of S phase is 6 to 8 hours, in which histone proteins is also synthesized, as well as the genetic material, that is the basic part, means DNA, is synthesized in the S phase. The S phase is again followed by the third phase, that is known as the G2 phase. G2 phase is a second growth phase, or it is also known as the second gap period, in which the complete development of the cell is occur. The protein synthesis and RNA synthesis is occur in the G2 phase, as well as the metabolic processes is carried out of the cell. Means cells is completely mature or is completely developed and it is ready for the cell division. In the interphase cell, it is the resting phase in which growth of the cell is occur and the cell is ready for the next division or the further division. Along with the interphase, the cell cycle consists second phase, that is the M phase. M phase, M is stand for the mitosis and meiosis. M phase is mitosis or meiosis. Both phases, means mitosis or meiosis, carried out by two processes. One is the karyokinesis and another is the cytokinesis. Means the cell division is carried, division of the nucleus as well as division of the cytoplasm. In the karyokinesis, the both phases, karyokinesis and cytokinesis, are present in the mitosis as well as in the meiosis. In the karyokinesis, the division of nucleus takes place. Division of nucleus takes place and two daughter nuclei are formed in the karyokinesis. And it is followed by the cytokinesis. In the cytokinesis, the division of cytoplasm takes place and two daughter cells are formed from the parent cell or from pre-existing cell. The both phases occur in the M phase. Now we move on our topic, that is the types of cell division. Types of cell division. In the cell division, there are three types. There are three types. First one is the amitosis. Amitosis, it is very simple type of the cell division. It is the direct division. Direct division of the cell is carried out by the process of amitosis, and it is very simple type. The second one is the mitosis. Mitosis is known as the indirect division. Mitosis is known as the indirect division. It is also known as equational division. It is also known as equational division. The haploid cell as well as diploid cell, both are divided by the mitotically. Means the mitosis occur in the haploid cell as well as in the diploid cell, and it is known as the indirect division. The third type of the cell division is meiosis. Meiosis is known as the reduction division. The meiosis is known as the reduction division. The cells are diploid, which undergoes the process of cell division is meiosis. Means the diploid cells are undergo the cell division is meiotically. That's why it is known as the reduction division, because the cells are halving in the daughter cells. Means the chromosome numbers are halving in the daughter cells than the parent cell. Now the types of cell division, we discussed about the first one, that is the amitosis. Amitosis is very simple type of the cell division. It is known as the direct division, in which it is simplest mode of the cell division. It is known as the direct division. It occur in the unicellular organism as well as fetal membrane cells. It occur in the fetal membrane cell, in the unicellular organism, and in the abnormal cells, in which the cells are elongated and constriction takes place between the cells, and these are separated. From that, constriction is deepened and the two cells are formed from the single cell. It is occur in the unicellular organism, and it is very simple process of the cell division, that is the amitosis. Means amitosis is direct division. There is no subphases occurs in the process of amitosis. Okay, so next type of the cell division is mitosis. The next type of the cell division is known as the mitosis. It is also known as the equational division. It is also known as the equational division because two daughter cells or two cells are formed from the single cell, and both cells are similar to their parent cell. Both cells are similar to their parent cell, means these are the identical to their parent cell. When the cells are divided mitotically, the daughter cells are identical to their parent cell. The mitosis, again, it has two steps. One is the karyokinesis and another is the cytokinesis. One is karyokinesis, means the division of nucleus, and it is followed by the cytokinesis, that is known as the division of cytoplasm, and lastly the formation of two daughter cells. Here, first we discussed about the karyokinesis. So karyokinesis means nuclear division, or the division of the nucleus takes place in the cell, that is the karyokinesis. Again, the karyokinesis has four subphases. The karyokinesis is divided into four subphases. First one is the prophase. In the prophase, the chromatin network is condensed, means the condensation of chromatin network is carried out in the early prophase, and the migration of centromere also takes place, as well as appearance of the mitotic apparatus and disappearance of the nuclear membrane. At the late prophase or at the end of the prophase, there is completely disappearing of the nuclear membrane, means nuclear membrane is completely disappear, as well as the nucleolus which is present inside the nucleus, it is also disappear, and the centrioles are again start to move from each other. The centrioles are trying to move from each other at the end of the prophase, and at the end of the prophase it is followed by the metaphase. In the prophase, means in the end of the prophase, the nuclear membrane is completely disappear, as well as the nucleolus is also completely disappear, and in place of the nuclear membrane there is the formation of kinetic spindle, or the formation of spindle fiber takes place. So in the metaphase, the prophase is followed by the second phase, that is the metaphase, in which the kinetic spindle, or we can say that the spindle fiber is appear. Okay, and on that spindle fiber the chromosomes are arranged. The chromosomes are arranged with sister chromatids. At the end of the prophase, the chromosome has sister chromatids, and these are arranged at the equatorial of the spindle fiber, and the chromosomes are arranged in the middle plate. These are arranged at the metaphase plate, and these are arranged at the middle of the means at the equatorial, and it is the middle. That's why the phase is known as the metaphase. The kinetic spindle or spindle fibers are present in the next phase, that is the anaphase. But before the anaphase, in metaphase, here centromere is divided. Centromere with chromatids are present in the chromosome. These centromeres are divided into two, and the distinct chromatids are present to respectively centromeres. In the anaphase, there is the daughter chromosomes are formed. Daughter chromosomes are formed from the single chromosome. There is the division of centromeres takes place, and the daughter chromosomes are formed, and the daughter chromosomes are pulled away to each other. Okay, the daughter chromosomes are pulled away to each other, and they are arranged at the respective poles. They are goes away from each other, and they are arranged at the respective poles in the anaphase stage, and they are also interconnected by the interchromosomal fiber. They are interconnected by the interchromosomal fiber, but they are pulled away to each other, means they are pulled away to opposite to each other with respect to poles, respect to their poles. The anaphase is again followed by the last stage of the mitosis, that is known as the telophase. That is known as the telophase. In the telophase, what happens? The kinetic fiber or spindle fiber is absorbed by the cytoplasm. The cytoplasm is completely absorbed what? The spindle fiber or kinetic fiber. In the telophase, the nuclear membrane is reappear, as well as the nucleolus also reappear. The nuclear membrane as well as nucleolus both are reappear, and the nuclear is completely divided into two in the telophase. The nuclear is completely divided into two, which nucleolus, nuclear membrane, and respective daughter chromosomes. Here the telophase, in which both nuclei are separated, means these are known as the daughter nuclei. These are known as the daughter nuclei, and after the telophase there is the cytokinesis. It is the last phase of karyokinesis, in which two daughter nuclei form, which nuclear membrane reappear, nucleolus also reappear, and the kinetic fiber or spindle fiber is absorbed by the cytoplasm. We can say that it is mixed with the cytoplasm, and the formation of two daughter nuclei. The telophase is followed by the cytokinesis. At the end of the telophase, karyokinesis is over, and it is followed by the division of cytoplasm, that is the cytokinesis, in which the constriction takes place between the cell or in between the two nuclei, and formation of two daughter cells by the division of cytoplasm. By the division of cytoplasm, two daughter cells are formed with nucleus, means nuclear membrane, and nucleolus, and respective daughter chromosomes, and the daughter chromosomes are equal in number to their parent cell. Means the daughter chromosomes are equal to their chromosomal number with respect to their parent cell, and here the process of mitosis is completed. The significance of the mitosis is what? It is known as the equational division. It is known as the equational division because the chromosome number is same as their parent cell, and all body cells or all somatic cells are divided by the process of mitosis. By the process of mitosis, and the formation of two daughter cells are formed at the end of the cytokinesis. Again, in the mitosis, the equal distribution of equal distribution of the genetic material. Means in the daughter cells, the DNA are equally distributed in the both daughter cells, and hence it is known as the equational division, and the mitosis is help in the growth and the development of the organism. It is help in the growth and the development of the organism in which the chromosomal number is constant and it is maintained by the process of the mitosis. Okay, thank you.